Good afternoon, everyone. My name is Dr. Nishamol P, and I am delighted to present our work entitled Explainable Multimodal AI for Drug Repurposing, Integrating Multi-Omics and Clinical Text for Precision Oncology. This presentation is part of aiHealth 2026 conference here in Mytilene. Thank you for being here. This work addresses one of the most important challenges in AI-driven healthcare today, that is how to combine diverse biomedical data sources into a single intelligent framework while ensuring that the model remains transparent and clinically interpretable. As we know, the modern oncology generates enormous volumes of genomic, transcriptomic, and clinical information. Although the artificial intelligence has achieved remarkable predictive performance, clinicians often hesitate to trust the black box systems. So, therefore, our goal was not only to improve the prediction accuracy, but also to make every prediction explainable, allowing clinicians to understand why a particular drug recommendation is made. During the next 10 minutes, I will briefly discuss the motivation, methodology, the major findings, and future direction of this research. The cancer treatment is becoming increasingly personalized. Every patient has a unique molecular profile, clinical history, and disease progression. However, these different data sources exist in different formats. As we know, the biomedical data is incredibly complex. We are dealing with genomics, transcriptomics, proteomics, plus unstructured clinical text. Each modality has its own structure and noise pattern. The multi-omics dataset include gene expressions, mutations, and molecular biomarkers, while the electronic health records contain valuable clinical narratives written in natural languages. So the traditional machine learning methods often analyzes these modalities independently, resulting in the loss of many important biological relationships. So at the same time, many deep learning models provides accurate predictions but cannot explain their reasoning, which limits the clinical adoption. Therefore, the objective of our work here, first to integrate multi-omics data and clinical text into a unified AI framework. Second, to improve the drug repurposing prediction performance. Finally, to provide an accurate and explainable predictions that clinicians can interpret and trust. Several landmark studies have demonstrated the transformative potential of AI in medicine. Eric Topol highlighted the convergence of human expertise and AI in healthcare. The Landberg and Lee introduced SHAP, which has become one of the most widely accepted methods for interpreting machine learning predictions. Attention mechanism introduced by Vaswani and colleagues revolutionized the natural language processing and now form the basis of modern transformer architecture. Likewise, AlphaFold showed how deep learning could solve extremely complex biomedical problems. Despite these advances, relatively few studies combine the structured molecular information with the unstructured clinical narratives while simultaneously maintaining explainability. This gap motivated our proposed multimodal framework. This slide summarizes the overall workflow of our proposed framework. We first collect the data from multiple sources, including multi-omics dataset and clinical text records. Before model training, all the datasets undergo pre-processing. This includes normalization, quality control, feature engineering, and temporal alignment in order to ensure that corresponding molecular and clinical information belong to the same patient. For the structured omics data, we use convolutional neural networks to learn high-level biological representations. For clinical narratives, transformer-based language models such as BioBERT generate the contextual embedding from unstructured medical text. These learned representations are then combined using an attention-based multimodal fusion mechanism. Rather than treating every modality equally, the attention mechanism automatically learns which information is the most relevant for predicting effective drug candidates. Finally, the integrated representation is used to predict drug-disease associations, and the model performance is evaluated using cross-validation together with the metrics including ROC, AUC, F1 score, and accuracy. Our experimental evaluation focused on squamous cell carcinoma using approximately 500 patient samples from publicly available TCGA HNSA datasets. We integrated multiple types of molecular information, including gene expression, profiles, and mutation data. These structured datasets were complemented by unstructured clinical narratives obtained from the medical records. Integrating the structured biological measurements with the free text clinical descriptions enables the model to capture the complementary information that would otherwise remain isolated. The results demonstrate that the integrating multiple modalities consistently improve the predictive performance. Compared with the models trained only on genomic information or only on clinical text, the multimodal framework achieved substantially higher ROC, AUC, and F1 score. Overall, we observed the approximately 10 to 13% improvement in ROC, AUC, and around 8 to 10% improvement in F1 score. More importantly, these improvements indicate that multimodal biomarkers and the clinical narratives provide complementary information. By combining both sources, the model gains a more comprehensive understanding of tumor biology and patient-specific characteristics. One of the major contributions of this work is explainability. Instead of producing predictions without justification, we applied SHAP analysis to quantify the contribution of individual molecular biomarkers and the clinical features. Attention visualization further reveals which modalities the model emphasizes during prediction. This provides valuable biological insights by identifying influential genes, pathways, and clinically relevant terms associated with therapeutic responses. Importantly, many of these findings are consistent with existing biological knowledge, increasing the confidence that the model is learning meaningful clinical relationships rather than the statistical artifacts. Such explainability is essential for the future clinical decision support systems. Like every study, this work has several limitations. First, the dataset size remains relatively modest and focused primarily on squamous cell carcinoma. Therefore, larger multi-center validation studies will be required before the clinical deployment. Second, additional modalities such as medical imaging, proteomics, and longitudinal patient monitoring could further improve the predictive performance. Finally, multimodal learning introduces significant computational complexity and requires careful harmonization of heterogeneous datasets. In future work, we plan to expand this framework across multiple cancer types, incorporating imaging data and evaluate its performance in real-world clinical environments. In conclusion, this work represents an explainable multimodal AI framework that integrates molecular data and clinical narratives for precision oncology. The proposed approach improves the predictive performance while simultaneously providing transparent explanation that clinicians can understand. Explainable AI represents an important step towards the trustworthy clinical decision support because healthcare professionals require both accurate predictions and understandable reasoning. Ultimately, our long-term vision is to develop clinically deployable AI systems that support personalized treatment selection and accelerate drug repurposing for cancer patients. These are the key references that influenced our work, including foundational studies on explainable AI, transformer architecture, and AI application in healthcare. I would like to acknowledge the researchers whose work has contributed to this study and also thank the organizers of aiHealth 2026 conference for the opportunity to present. Thank you very much for your attention. I will now be happy to answer any questions. Thank you.